I didn't push record, though. So I asked you the question, what controls gene expression? So how is gene expression controlled? I see alleles. Well, alleles are the versions of the genes that we are controlling. That is like a smart, I don't know. Right? Gene expression, IDK. Oh, come on now.
Good morning, John Glenn High School.
Not sure.
Happy Thursday.
I like promoters.
Today is our first student of the month.
We'll talk more about that.
I'll be announcing the staff members of the marketing period later.
Emotions and smiles, serotonin, that's a protein. Yes. Smiles, hugs, chocolate. Yeah.
If I call your name, please come down to the common.
Is Mr. B taking people right now?
April Primer.
Oh, is this student of the marketing period day?
Brendan Levasseur, Hunter Kukla, Jordan Bird, Jordan Hinton, Max Vipke, Noah Thalen, Shayla Connor, Tabitha Burnett, Tiffany Bessel, Trinity Sewell Watson, Alex Martek, Alexandria Martek, Anthony Strickland, Brady Race, Brooke Cunningham, Ian Woodbury, Jacob Sanders, Jonathan Horrigan, Jack, Katie Johnson, Chloe Lang, Michael Maldonado, Noah Hill, Sadie Treichel, Seth Goss, Skylar Schmidt, Jennifer Smith, Jill Chattal, Chantal Piggott, Elizabeth Garcia,
I'm selfish. I keep going.
Jennifer
I forgot. I've been gone for 2. Otherwise, they'd have been on there. I'd have put all you guys on there. Usually what I do is...
Logan Miller, Nick Carpenter, Olivia Nicholas, Parker Jeske,
So like fourth marking period, you guys will all get together.
Brittany Keeler, Brooke Hout, Drew Pomeroy, Jesse Ballard.
If you're listening to the YouTube recording, we are waiting for the marking period, student of the marking period list to end. It will go forever. It's always on YouTube. There's a playlist. Yes.
Tyler Monville, Tyler White.
Is that it?
Come on down to the
I will put you guys on, all of you, fourth marking period. I promise. I do it all at once. Okay. Yes. I have you all pick a word from there, that list up there, and we all come up and you fill it out because you are all beautiful students and you're perfect, I know. And so I just do it all at once so our class is disrupted on only one day and we all get to go and then we come back. So, well, that would just be abusing the system. So today we're going to be looking at gene expression. So the words, I was looking for like maybe gene regulators, operons, operators, inhibitors. Those would have been the answers I was looking for, but I know we haven't learned this yet. I'm trying to ignore you, but it's not working. Yes, I know. So we're going to be looking at how we control the regulation or the expression of proteins. Because my eyes are brown because at a certain stage of development, it was after I was born actually, because my eyes were blue when I was born. My body started telling me to make those brown proteins, and my eyes turned brown. Somebody gives you a hug, and your brain releases this nice dopamine chemical in your brain, which makes you release another chemical called serotonin. Those are all proteins, you guys, that are activated by an actual chemical. And we're going to learn a little bit about how that works this chapter. How do you activate a gene? We'll talk about the common form of gene regulation in prokaryotes. It's a lot simpler than ours. I'll give you a hint. You've already done it in lab. When did we already look at the gene regulation in lab? What would have been something that activated the gene in lab? What would have been something that activated the gene in lab? And yes, I want 26 answers. What would have been something that regulated a gene in a lab we've done so far this year? Pick something that regulated a gene. Turned it on or off? Yep. What would have activated a gene to produce a protein, hence expression? Keep calm and apply to college. That's a good bookmark. Yep. Is that yours? Oh, I just threw something dirty on your bag? Sorry. Well, don't throw it back on the floor. I'm waiting. You guys, we will not continue until I get full participation. Take a stab at it. Well, take a try. Shouldn't say stab, shoot, punch in school. All those things are bad. Come now. Let's go. Oh, that's quite the answer. Arabinose for sure, right? Arabinose is what turned that gene on or off. Heat shock helped us take in the plasma, but didn't cause the gene to be expressed. UV light was more of a reflection of the gene. That's like saying light causes my eyes to be brown, which kind of true, I guess. But who is Alexi's birthday?
That's right. And you didn't take me down to that breakfast. I'm just saying.
This whole class deserves that celebration for Lexi's birthday.
We could have had Lexi's birthday party down at the Commons. I know, right? Piglo and a Ravenose. City. What is that? Sidcat? Drake, are you calling me a liar? I ain't calling you a truther. What is that? I don't know. Drake? No. The Ravenose sugar caused the Piglo gene to be expressed by the plasma. Yes. Okay. All right. Oops, I got to go back. We're not done yet. So we're also going to look at how gene regulation in eukaryotes is different than that in prokaryotes. You guys, what is our DNA held inside of? A nucleus. So ours is not directly influenced by the environment. Now we'll also look at why proteins are so versatile and why we have so many different types. Folks, get focused on your notes. I'm not going to give you all your notes. Okay, we're gonna jump into karyotyping, but we have to get to like a stopping point today. So scientists have learned that gene expression, transcription through translation. Again, if I ask you what gene expression is, you know I'm talking about transcription, copying DNA and RNA, and translation, translating that information out of the ribosome into amino acid language. Now, not all genes are expressed in every cell, nor are many genes expressed all of the time. So I don't always release insulin and make it in mass quantities. I only do that if somebody like pulls out a knife or a gun or makes me feel really nervous, right? Then you release this chemical. Anybody ever have to public speak in front of a group of people? Yeah, I did it 2 days ago. Do you guys think I got a little bit maybe shaky and my voice quivered a little bit, stuff like that?
No, you're awesome.
Well, I am with you guys. I see you every day, right? Kids don't scare me. Guess who does? People my own age, right? And so even to this day, when I get up and have to talk in front of a group of people that are equals to me, you know, it is a little... No. That was terrible. That was like a North Korean statement right there. Let me retract that whole thing. I just hurt all of your feelings. I'm not doing good today. All right. So I would not be like, my name didn't get called for staff remarks.
You don't vote for us, we don't vote for you.
So, all right. So listen, here's the thing. When I get in front of people, I get nervous sometimes. It doesn't happen all the time, but it does. It did happen a couple days ago. I was in front of like 300 people at a Google Summit talking. That makes you nervous, right? And so I got sweaty, right? You could hear probably a little quivering in my voice. What's happening is my body, it goes into like that prehistoric flight versus flight mode, and I can either run or fight. And the only problem when you're stuck talking in front of people is you can't do either, right? So you just stand there, and all of that adrenaline goes to all your muscles because your body is telling you you're really scared. You're releasing all of this protein called adrenaline. It's affecting everything in your body, making all of your blood vessels dilate. You're getting way too much blood. Your body's overheating. It's kind of weird that your body does this, but that's the way we evolved, right, to run from big, large animals when they scare you. And so we didn't evolve talking in front of 300 people. And so our body doesn't understand that. So it releases this protein. Gene expression is happening in mass quantities, and you hear it and see it in your phenotype, right? Your genotype gets expressed. So not all genes are expressed in every cell, nor are many genes expressed all the time. Here's the thing. You have 30,000 different genes in all of your cells. Skin cell, muscle cell, brain cell. They all have the same DNA. Why are they different? Let me ask you that. Why are they different? I can adjust that. They're good now or bad now? Okay. I did change them a little bit. What? What? Why are our cells different? They all have the same DNA. Why are they different? Why are our cells different? Ugh. Try to spit all that out. Why are our cells different? They have the same DNA. Why are they different? Come on now. I don't know is acceptable, but I want 26 responses, please. IDK is acceptable if you don't know. But please, give me an answer. I want 26 people participating. I thought I had 26. I had 26. There were 26. Yeah, there were 26 turned on. Oh, one might be in the potty or something. Come on now. Be the little choo-choo that did. That could. Isn't that what it is? The little engine. That's a little choo-choo. Okay, so they can do different jobs. They're specialized. Different genes are expressed. That's a good one. Different genes. You know, every cell has the same DNA. They just have different genes they're expressing, which makes them specialized for sure. Who knows? I do. You do. You're gonna. All right. They code for different proteins. Yes. Kind of. They all have the same protein code there. Specialized jobs, different codons, transcription functions. Okay. Different responsibilities, environments. They are in different environments. Each cell has a different job. Yeah. Okay. Gene regulation allows only one necessary protein for the specific cells to specialize and effectively work together as tissues, organs, and organ systems. Okay. Our cells are regulated to do special things like okay. So here's the thing, guys. Oh, I'm sorry. Where is it? Is it up here? Okay. So here's the thing. Our cells, like our skin cell compared to our muscle cells, they have the same DNA. I can actually take your skin cell out, take the nucleus out of it, put that in an egg, and it will make another you inside of a female. It's called somatic cell nuclear transfer. And we're going to be, hey, you guys, we are going to be learning about that very quickly here. We're going to watch a Harvard lecture together, and we'll stop and talk about it. But you guys, listen carefully. Every cell has the exact same DNA in your body. The cool thing is your cells turn different genes on or off. Because if you made them all at the same time, you wouldn't have enough energy to do it, number one, and your cell would have too many different proteins in it, and it would not function. You cannot support 30,000 different proteins. So you only have a few thousand that are turned on, usually, in each cell. So cells have complex systems that regulate whether or not specific genes are expressed, and expression depends on the cell's needs and the environment. What does your skin cell need? Well, we know it needs keratin to waterproof itself, so it makes the protein keratin. We know it needs to be flexible, right? Okay. So it makes the protein elastin. It has to be strong enough to not like push your finger through, so it makes collagen, right? Okay. Sorry. Now your muscle cells, they're inside your body. They're already protected. They don't have to be waterproof, so they do not make keratin, right? They make actin and myosin, which are contracting contracting protein fibers. And yes, we will deal with that a lot more in anatomy. You guys will be looking at a lot of these things under microscopes in the second semester. Now, through gene regulation, a given genetic sequence can be expressed in different ways and in different bodies or tissues under different conditions or at different times. Now, I've given you a few different cell types. I've talked about what happens when you get scared. You release insulin and make a lot of that. So it's not just the specialized cell and where it's at in the body. It's the conditions of where you're at. Okay? What does that mean? That means if I go to Florida and I lay on the beach, my body's going to make more melanin, right? That's the protein that makes you tan. Also, a good indicator if you're exposing yourself to too much sun and causing cancer. So gene regulation accounts for changes during development as well as differences among organisms that have similar genes. Now I want you to think about this. Let's rewind the clock and just don't talk to your neighbor for a moment and think about back to when you were just a single cell. Okay? I can barely remember it. Okay, so your dad's sperm and your mom's egg came together and they made you. Okay? You guys, don't talk, just listen for a moment. I promise I'll let you talk in a minute. But there's a single cell, and it doesn't know what to do. It has no clue. Who tells it what to do? Who tells the cell what to do? You got one zygote. We all started out as a zygote. Sounds like an alien. We all started out as a diploid, fertilized sperm and egg, two gametes coming together to make a diploid zygote. What in the world tells that single cell what to do? Let's see if you guys... Okay, yeah, your mom. I'm not going to have you type that in. So your mom. All right? So your mom has been telling you what to do, right? We all know moms tell us what to do. But her body is actually telling that cell what to do. It's saying, cell, divide rapidly. It's saying, okay, cell, it's time to form different types of cells. You need to actually start forming cardiac and nerve tissue first. That's the very first thing stem cells do. They'll start to pulsate and beat. That's creepy. Okay, they will do that before you have a nervous system. So the very first cells, they start to throb like this, and they pulse. Your heart is an independent organism from your whole body. You guys know how you can take a heart out, restart it, put it in someone else? It's an independent tissue. Okay? What? Okay, something like that. So your heart, you guys, you can cut a turtle's head off, and its heart will beat for 6 hours on its own. I've done that lab in college. It's fun, but a little inhumane. We aren't going to do that in here. I'll get too many parent phone calls. But here's the thing, you guys. The very first tissue that becomes differentiated is made of many different tissues, and the first one is cardiac tissue. Okay? And so your mom must have sent signals from her brain, and these signals are called transcription factors. What are they called? Transcription factors. What is transcription when you copy DNA onto RNA? So your mother's brain sends proteins and hormones called transcription factors in an orchestra to her uterus, which is where your zygote would have been. And then that zygote starts to do some very strange things. It starts to divide at different rates. It starts to develop and make different proteins based on what mom's signals are sending. And so you have this like symphony of what to and not to do going to these cells all the time during 9 months of gestation. Okay? Do you think there's a lab anywhere on planet Earth at this time, I say at this time because someday there probably will be, that can replicate that? No. No. There's not a computer built yet that can handle that complexity. They're working on it, but it's not there yet. So what you guys need to understand is, yes, we can clone people. We can clone animals. We can clone them right now. It's no big deal. I take your skin cell out. I take the nucleus out. I put it in. Let's say I grab a random female, and I take her egg out. I pull her nucleus out of that egg, and I shove your nucleus from your skin cell in her egg, and I embed it in her uterine wall. She will have another you. That's true. That happens. That's called somatic cell nuclear transfer. You guys are going to learn about that. That's how they clone Dolly the sheep. They've been cloning frogs since like the 70s. Okay? You can, but it's illegal in the United States. I'm sure somewhere they've tried it and done it. I don't know where. Ethically. Remember bioethics? We don't. It's illegal right now. Okay. I'm actually okay with it. I don't care. But that's me. Yeah. You have to vote on things like that, right? What? I have to check. I'm sure somebody's done it in the lab. They might not have like published it outright, but okay. So one benefit of gene regulation is that cells can use energy and materials efficiently. So like imagine your cell trying to make all 30,000 proteins all at the same time. It's not going to function. It will not be able to support all those different proteins. Like your muscle cells, they have like just a few basic proteins actin and myosin, two of the major ones, if they were making keratin too and trying to export it to your skin, that's too much energy. Okay? You would not have enough energy to go through contraction and relaxation. So you have to only make the cells you need in specialized differentiated cells. Now, nearly every step in the process of gene expression can be regulated or controlled. And we need to regulate gene expression. If we don't, we get proteins in the wrong place. And if we get proteins in the wrong place, sometimes that can lead to, let's say, scar tissue in your lungs, and then you have a serious problem. That's kind of like cystic fibrosis, right? Now, a molecular system that controls the expression of a specific gene is called a genetic switch. Why is it called a genetic switch? Because it turns the gene on and it turns the gene off. Now, often the switch is triggered by factors or conditions outside of the cell. What do we call those factors? See if you guys remember. What do we call those factors that control gene expression? This will be on your test. What do we call the factors that control gene expression? I just had you all repeat it a little bit ago. What do we call the factors that control gene expression? What do we call the factors that, in IDK is an honest answer, but I want answers from everybody. Okay? It is okay to be wrong. No more Drake quotes of the day, though. Drake is a male duck. Wah, 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 wah. It's a Drake. It's a duck. It's a male duck. Oh, geez, I just quacked on YouTube. That's not good. I recorded myself quacking. Do you know what Drake puts in his soup? Quackers. Sorry. Sorry. I apologize for that joke. Yeah. That's how my brain works. I know, it's weird. It's like the Laffy Taffy dictionary rolling in my head all the time. All right, let's see what you guys came up with. There you go. Transcription fact. Random dancing, no. Transcription factors. Yay, I had a couple right answers. Something transcription. That's okay. Hey, if you got transcription out of that and you didn't have factor, that's okay. I don't even know what that is. You know, I know who you are. Okay. I like it. Okay. You ask a lot of questions, Mr. Mason. All right. I do. That's my job. I get paid to do that. All right. So the major form of, it is transcription factors, guys. The major form of gene regulation in prokaryotes depends upon operons. Operon, operon, potato, potato. Everybody say operon. Okay. Say operon. Say operator. Say operons operate genes. For bacteria. Okay. Yeah. So an operon is a gene regulation system in which adjacent DNA segments control the expression of a group of genes with closely related functions. You guys already did this in lab, believe it or not. Now, operons are common in bacteria but uncommon in eukaryotes. They do exist. The only problem is our DNA does not have this direct contact with our environment because we have a nucleus. So a bacteria has no nucleus. Therefore, bless you, their DNA is in direct exposure to whatever their environment is. Oh, I'll go back. Okay. I'm sorry. I'll work on that. If you have extra time, I'd love to have you come in and you could go through and highlight all those words for me. I'll make you do it. You come in at lunch, I'll let you do that. Oh, for all of them? Do you know how many, for every single? Okay, oh. Who's Drake? Yeah, right. All right, so bacteria are single cells that must get food directly from the environment. So if you were our E. coli, you were after arabinose, right? So given a stable environment, a bacterium will need a steady supply of proteins and will tend to keep expressing the same genes and in the same way if the environment's stable. If the environment's unstable, it will change. In our lab, when we did the E. coli transformation, the E. coli grew when arabinose was present, but then it didn't when it was absent. That would be a changing environment. Now, usually in the bottom of the ocean, there's a dense supply of phytoplankton, and they always have arabinose. So those jellyfish are like always glowing. So that's a stable environment. But if arabinose were like washed in and then washed out with phytoplankton, then that would be an unstable changing environment. So if the environment changes, a cascade of changes in gene expression may result, and in a way, the environment flips a switch. Put arabinose in the environment, flip the switch on. Take arabinose out, flip the switch off. That is gene expression. That is a form of controlling a gene. Now, this lac operon that we're talking about, or operon, I don't care which one you say, is an example of gene regulation and is found in the bacterium E. coli. In fact, it is the same mechanism that we were looking at under the UV light. Now, usually when you eat or drink a dairy product, the chemical lactose, milk sugar, is digested by E. coli cells living in your gut. These cells can use the lactose for energy or for other needs. Here's something really cool about all of us. We are all lactose intolerant. You guys only produce the enzyme lactase until you're almost a year old. Then you don't make it anymore, and you can no longer digest milk. And you all look at me like, that's not true. Because I drink milk. I put it on my cereal. And I would tell you, yes, but you have a culture of E. coli living in your lower gut, part of your intestines, that actually breaks down the lactose for you. And then it gives you the glucose. Then you go through glycolysis. And you guys remember all that electron transport chain? Okay? Maybe. What? Oh, yeah, you can't. You'll throw it up, yeah. So, like, here's the cool thing. You guys, some of us fight off this E. coli, and we can't host this in our body. Then you would said to be what? Lactose intolerant, okay? So the people that are truly, and we are all lactose intolerant, but the ones that are labeled lactose intolerant do not house a healthy enough culture of E. coli to break it down, okay? So at first the cells must attach to absorb and then break down lactose. These tasks require 3 different enzymes, each of which is coded for by a different gene. And this lac operon is what we're going to be looking at. Now, the system that involves the lac gene is called the lac operon. This system includes the 3 genes plus a promoter site and an operator site. The promoter will promote the transcription of the gene. Remember, that's where we start reading DNA, the promoter site. The operator site controls if we read it or not. That kind of makes sense, right? The operator. So when lactose is available, the system turns on. So if arabinose is present, we see the gene being expressed. And the 3 genes are transcribed. Now, when lactose is absent, the system turns off, and transcription is blocked. Basically, it's like putting a screen or a blind up in front of a page. You can't read it. Okay? Once you can't read, you can't transcribe. And at that point, transcription is stopped. Remember, transcription is the first step of gene expression. If you don't transcribe, you do not get a protein, period. You need RNA. So it looks, it's blurry. It looks something like this. You have a regulator gene. This gene codes for this specific repressor protein. We call it a repressor protein because if it binds to the operator, the promoter cannot be read. The promoter cannot go past this point. Now here's the question. If you look over here, 1, 2, and 3, these are structural parts of the gene. We're going to talk more about introns and exons in a bit. This is probably this is one of the most important things in this chapter for us all to understand. This part right here is what will code for the protein. This operator controls whether or not we can read and copy this DNA onto RNA. If the repressor protein binds to the operator, what enzyme, now think very carefully, what enzyme cannot continue to transcribe DNA? What enzyme cannot continue to transcribe DNA if that inhibitor is there, blocking that operator and the promoter? If there's a repressor protein binded to the operator, which enzyme cannot do its job? I'm forcing you to go back to chapter 13 in your head right now. This needs to be common knowledge in here. This cannot be something you forget. This is not one of those classes where we only do 3 chapters the whole year. You have to remember everything the whole year. Well, semester. See what you guys are coming up with. See what's going on. Get rid of the Drake quotes. Oh, yeah, polymerase. That's what we're looking for. Not DNA polymerase. We aren't copying DNA onto DNA, right? We're copying DNA onto RNA. So the best answer here would actually be RNA polymerase. Oh, there's one. Okay, good deal. What, did I miss something funny with it? Oprah's on. Oprah on. No, sorry. Okay. So looking at this lac operon, it was RNA polymerase that'll be right here, ready to copy that gene. And as soon as this repressor protein leaves, it will transcribe the gene. But if this repressor protein is there, it's like a roadblock. Nothing's going to get transcribed. Now, I know we've been going for a little bit here. I told you I was going to get to karyotypes. So what I'm going to do is stop. We're going to review this tomorrow to start class off, or Monday, because this is an extremely important thing, and I don't want to just blow through it. I want it to be fresh in your mind at least 2 days.